Okay, here are the directions for today's lab. I'm not going to give written instructions. They will only be video style here and now. Luckily, I put this on YouTube and put the link in the classroom, and that's why you're watching it now. And you can go back to it multiple times, okay? Let's start first. The problem. We're trying to figure out if yeast prefer different types of food. Reality, we know that yeast will ferment through respiration sugars, and as a byproduct, they make carbon dioxide. So what we're going to do is we're going to feed yeast, the same yeast, different types of sugar. And so when we think about variables again, right? Sugars, or the type of sugar, will be our manipulated independent variable, our mix variable. Now, what do we know about yeast and sugar respiration? They make carbon dioxide gas, which cause our little baggies to fill up. So we're going to take that knowledge and we're going to say, okay, we're going to base how well yeast like certain sugars based on how much carbon dioxide they make. The more carbon dioxide they make, the more they dig that sugar. Or maybe just the easier it is for them to use that sugar. All right? So let's look at the different types of sugars, right? Our mix variable, different types of sugars. So here are the different sugars in these small New York State issued cups, like molasses or corn syrup or powdered pure dextrose. And each sugar will have a spoon in it because it takes a spoonful of sugar to help the medicine go down. Each lab group is going to need one beaker, and one test tube. One beaker, one test tube. That's what each lab group is going to need. I have 12 beakers over here, but what I want you to do is, there are a couple of doubles. Make sure that before you use a double, all the singles have been used. For example, like there's 2 glucoses, right? If they're not 12 lab tables, then only use one of the glucoses. And then I need one group to do the control. That's a group that knows that they're probably not going to get any result, but has to figure out, what are you actually going to put in here? All right, so what do you need to do? The first thing you need to do is you got to take your beaker, right? It's labeled with the type of sugar, syrup, and we're going to add water to it. We're going to add 100 milliliters of water. Now, these are 250 milliliter beakers, 250 milliliter beakers, but they're usually graduated. Here it says 100. So we'll just fill it up, and we can just eyeball that amount. To be honest, being perfectly there is not necessarily the be-all, end-all. That's number one. Number two, they say a spoonful of sugar helps the medicine go down. So now what you're going to do second is you're going to take a spoonful of your sugar, in this case syrup, which is not very easy for me to do with one hand, but spoonful of syrup, and then we're going to dissolve it in our beaker. So I've got 100 milliliters of sugared up, syrupy sugar water. All right, stir it up nice and good. Now, previously in Mr. Kalbeck's class, we learned that yeast work best when they're in an environment that's warmer, especially up to about 90 degrees Celsius. All right, so once you've put your sugar into the water, you're gonna leave it here on this heater for, let's go at least 5 minutes, so that give it enough time for this solution to get warm, right up to warmth, 5 to 10 minutes or so. Then, the fun part here is I have yeast solution, right, or yeast, powdered yeast. You're gonna take 1 spoonful of yeast and put it in your beaker. 1 spoonful of yeast, put in your beaker. Then, stir. Then you're going to take your test tube And let's see if I can do this in a way you can see it. You're going to take your test tube, and here's the test tube. Yeah, your test tube, and you're going to fill it with, fill it all the way up with the solution. See how I filled it all the way up? Filled it all the way up. And then here's the tricky part. We're going to try to invert this test tube into the beaker. Invert the test tube into the beaker. So I'm going to take my test tube, my beaker kind of sideways, and go whoop. So I have now a test tube upside down in the beaker. And what you're going to do is you're going to use a Sharpie marker. And I have my Sharpie markers, and you're going to mark the level of the solution in the test tube. And then we're going to let it sit. All right, and the idea is that as the yeast works, it's going to create carbon dioxide, right? So this bubble here at the top is going to get bigger. And then at the end of the period, we're going to measure how far the bubble gets. All right? Okay, so to review the directions, you get your beaker. You put 100 milliliters of water in the beaker. You add a spoonful of your sugar to help the medicine go down. You then heat your beaker over here in the hot plate for 5 to 10 minutes to get that solution warm. Then you add yeast, and the yeast will start to digest. You take that mixture of yeast and sugar and water, and you pour it into your test tube, right? You fill the test tube all the way up, and then you quickly invert the test tube into the beaker so it looks like this, and you mark with a line where the top of the solution is, and then we wait. Right? We've got a double period, so we're going to do this in the beginning of the period. So about an hour later, we'll go check. Now it's very important because other groups are going to be using this equipment when you're finished to give it a good cleaning. Right? Good cleaning means rinse, soap, brush the test tube brushes, and put all the stuff back so that the next class can do this as well. Mr. Kovac should not have to clean up after you. Okay? Hopefully you'll get some good results and we'll find out what kind of food yeast like best. All right, let's have fun. Okay, while you're waiting for your yeast to feed and make carbon dioxide, right, you'll open up this slideshow. So your lab report today is going to be to complete the slideshow, and it's called Respiratory Lab 2. And then on the second slide here, I've told you the directions, right? You need to include the following slides, right? Each slide, since it's a slideshow, it should be pretty looking and have an image too that goes with it. Slide number 1, what is the problem we're trying to solve? Hint, go back to the beginning of this video. 2, what are the variables? Hint, go back and look at the early part of this video. 3, how's the experiment set up? Hint, it's all in this video. What are the results of the experiment? Well, that's what we're going to do. We have to get at the end. So we won't be able to do slide number 4 until the end. And then number 5, you should be able to answer the question, once we get the results at the end, what type of food do you seem to prefer? Okay, so that's our lab report, right? That's what you're going to turn in. So as we're waiting for the yeast, you should get at least the first 3 slides set up. And then at the end of the period, I would say to give us enough time to clean up, 15 minutes before the period, you need to stop and then measure how big has the bubble gotten? Like how far has the bubble gotten the top of your test tube? And then share that with the rest of the class. Okay, because those are the results that everybody needs to have. Does that make sense? If it doesn't, raise your hand. I'll come over and I'll make it make more sense. All right, have fun out there today. Be safe. And remember, a spoonful of sugar helps the medicine go down.